Hello friends, today we are going to discuss the most fundamental and indispensable technique in molecular biology about polymerase chain reaction, PCR, polymerase chain reaction. Simply the DNA amplification. DNA amplification. PCR is nothing but in vitro generation of large quantities of target DNA. Large quantities of target DNA, and that means cell-free amplification procedure. Generally, we require, in general, there is a requirement of cell machinery to, that means what we call DNA duplication. But here we generate lump sum as many copies as we can as per our requirement with respect to the target DNA. That means, but it is a lab condition, what we call in vitro generation. Okay? And the most important thing is this PCR. Without PCR, most of the molecular biology techniques cannot proceed further. That is why I have specified and used the word indispensable fundamental technique. Okay. Regarding the basics, that is an in vitro generation of large quantities of target DNA. Okay, is the main objective of PCR. Carey Mullis developed this technique in 1984. Later, he got Nobel Prize. He was awarded with Nobel Prize for his discovery and for his work. And most important thing, the raw materials for PCR. I told you already. Large quantities, maximum, that means say n number of copies of the target DNA or the desired DNA. What we call the target DNA fragment. This is the first raw material, target DNA fragment. Then the basic bricks for DNA, DNTPS. That means all kind of 4 DNTP. That means DATP, DCTP, DGTP, and DTTP. Then primer, which is nothing but an RNA oligonucleotide strand. For example, you see 17 to 30 nucleotides. Then DNA polymerase enzyme. DNA polymerase enzyme. The polymerase, the word comes from the main role, the emphasized role of DNA polymerase. Actually, what kind of DNA polymerase does it involve? Initially, and earlier, clinofragment of E. coli we used to use, but the most important drawback with respect to clinofragment is it's like temperature unstable. That means it doesn't have resistance at higher temperature. Say at around 37 degrees centigrade is the optimum current temperature for this. But later onwards, the most what we call the breakthrough research is Thermus aquaticus, a archaebacteria, and the polymerase of the same, what we call Taq polymerase. Taq polymerase revolutionized the PCR, Taq polymerase, because it can work under stable conditions even at around 75 degrees centigrade, because it have the ability to resist to the higher temperatures. Because a kind of bacteria, Thermus aquaticus, the bacteria have the ability to resist the higher temperature, and so the enzyme is. That's it. So the basic requirements are like raw material. The target DNA may be say starting from 100 to 35,000 base pairs. It's somewhere in between 100 to 30,000 base pairs. The target DNA, the DNTPS, the primer, and DNA polymerase, and it will run in a machine, automated machine. Okay. So these are the raw materials. That means, and the basics. Okay. And how, and regarding its principle, what we call technique of PCR. For any molecular biology technique, there will be a principal procedure, what we call basic protocol. A basic protocol, just like the same here in PCR also. See, the principle of PCR. See, the first step is called denaturation. The second one, renaturation or annealing. And third one is DNA synthesis. Denaturation, say it will take place at 95 degrees centigrade. It will be around 55 degrees centigrade. Then synthesis around 75 degrees centigrade. Okay, these are the temperature zones. And one cycle of PCR takes some around 3 to 5 minutes. And this will be for around 1 minute. The same will be for 1 minute. And this is around 2 minutes. Okay. First we will discuss, then we will take a schematic presentation. I told you already, large quantities of target DNA. Say the target may be of around 100 to 35,000 base pairs. After we get the target DNA, it has to get denatured. Denatured means we have to destabilize the double helical structure simply by subjecting the target DNA fragment to the high temperatures of around 95 degrees centigrade. If we maintain the machine at 95 degrees centigrade, the DNA automatically will break down, forming single-stranded DNA fragments. The next step is what we call renaturation or annealing. This is like hybridization. Hybridization. Why and what exactly happens? It is with the help of a primer, an RNA primer. An RNA primer may be somewhere between 70 to 30 nucleotides. Just like the DNA duplication. But the thing is, in DNA duplication, there are two ways, leading and lagging. Just like the leading phase DNA duplication, the PCR runs. It is not discontinuous. It will continuously synthesize the DNA strand. Just like comparison, okay? And just like the DNA duplication along the leading strand, the PCR will run. And the same concept here, the primer will be required and it will initiate the DNA synthesis. But for that, the temperature has to come down to around 55 degrees centigrade. And after that, what we call annealing of primer. That means it maintain a complementarity. The concept is same. I'm not going to the base. Just like complementarity. Then after that, we require synthesis. What we call DNA synthesis, and this is very important step. Initially, and the first discussion I told you about polymerase role. Here, the DNA polymerase will that means extend the chain. Extend the chain means that extend from primer. From say for example, that means 3' hydroxyl free end of primer will be extended. That means what we call growing DNA chain. Okay, and it is done by the appropriate polymerase. Generally, we use stacked polymerase because it is stable at higher temperature for even 75 degrees centigrade. And the stacked polymerase will look off just like we will add the appropriate complementary bases, like ATCG. That's it, ATCG. That okay. And likewise, the DNA will synthesis. And these are the three phases. What we call totally one cycle of PCR. After that, we will go to the second cycle, third cycle. We can continue as many as we can. For example, at the end of 32nd round, we can get 1 million fold. That means 1 million identical copies of the target DNA at the end of the 32nd cycle. Okay, how? Okay, we'll discuss on by one. And these are the most important. The first, the denaturation, this renaturation, the DNA synthesis in one cycle, and the cycle will repeat. Cycle will repeat for n times, what we call. Okay, this is a basic principle for PCR. Okay, with regard to its representation, the first important thing is denaturation. Denaturation of target DNA fragment. Now how it works? Say denaturation. This is the target DNA. It has been denatured. That means at higher temperature, say 95 degrees centigrade. So two strands are separately formed. Okay, this is template 1 and this is template 2. Then followed by annealing, what we call primer. That means the primer will come and sit and will extend. Okay, just consider it as a 3 prime, 5 prime, and 3 prime and 5 prime. You know, we have discussed during DNA duplication, there is no DNA polymerase enzyme so far. Say for instance, be it is prokaryote, say in prokaryotes, DNA polymerase 3 will synthesize both in leading and lagging strand. Whereas in case of eukaryote, there are different kinds of DNA polymerase. DNA polymerase alpha, beta, gamma, delta and epsilon. Here the thing is, DNA polymerase delta in eukaryotes will synthesize the leading strand, whereas DNA polymerase epsilon will synthesize along the lagging strand. Then DNA polymerase gamma, it will synthesize the DNA in mitochondria, we call mitochondrial DNA duplication. What I mean the point it is, well, though there are different kinds of DNA polymerases both in prokaryotes and eukaryotes, but the common point is they will synthesize only in 5 prime to 3 prime direction. That means they extend the 3 prime hydroxyl group of primer. That's it. Because there is no DNA polymerase that can synthesize in 3 prime to 5 prime direction. That's it. That means there no enzymatic machinery to support the system along the, that means from 3 prime to 5 prime direction. Same concept of hierarchy. What we call, the primer will come here and it will be having 5 prime, and this is a 3 prime hydroxyl group. 3 prime hydroxyl group, okay? The same in here also. This is 5 prime and this is 3 prime hydroxyl group. After what we call prime, the second step, annealing. Then the third step, DNA synthesis. Simply, the chain will extend by DNA polymerase. That's it. The same concept. Say, here it is a primer and it will extend. That's it. These are called long templates. Long templates done by, it is again DNA polymerase will come into play. DNA polymerase is very important, okay? And this is what we call cycle 1. What are long templates? Long templates are nothing but, the DNA, the primer, it will lead to synthesis which is beyond the target DNase region. Beyond the target, sometimes will go beyond the complementary sequence of the second primer. The same, for example, for this is second primer, and the new DNA strand that is extending beyond the dots represent that it will go on beyond the complementary sequence of the second primer. Beyond the complementary sequence of the second primer, this is very important, because the target DNA region may go at just like extension out of the target DNA boundary. That is why they are called long templates, and they are also used as templates in the second cycle. Okay. In the second cycle, then they will be repeated. Cycle 2. The same concept. That means the original DNA strands and the long templates will be used here. This is the original DNA strand, and this is 1 and this is 2. This is a template, so this is the primer, and it will lead to another long template, and these are the the long templates. The thing is, it will generate short fragments. It will generate short fragments. You know, these are the long templates. Again, it will create long templates. These are the long templates, and these are the these are the short templates. These are the short templates. Why they are considered as short templates? Simply because it consists of DNA strand at one end. That means DNA strand with a primer at one end, with that of the sequence up to the second primer end. That means it doesn't go beyond the target DNA region. That's simple. It doesn't go. That means the short DNA fragment means that the short template means it doesn't go beyond the target DNA region. And in the next cycle, that means after next cycle, this short DNA is the most important part because it accumulates the target DNA fragments. That means the short DNAs are the key for our experiment. The rest, the long templates contain some extension nucleoside sequences, beyond the complementary sequence of the second primer. But the short contains the specific target DNA. Okay, this is second cycle, right? Then after that, the same concept, the third cycle will repeat. And who are the participants? The same concept. The original strand templates, the long templates, and the short templates. Same concept, That means, say for the DNA denaturation, annealing, and that means synthesis simply. It is called renaturation. Here also same. Repetition. Repeat. Then the third cycle. Here the three participants. Okay. This is the original strands. And this is another one long strand. And here the long strand. The short fragments. Then another one. This is a long template, and this is one, and this is short. And this is one more long template. Then this another one. To add confusion, these are the new. There's a new one. This is short. This is a long. This is a long one. This is a new one. New one. The short one. The long. Okay. That means the long one. The long one. Long. The short one. For illustration, we will take the short one like this. The short one. These are the short fragments. Short fragments, and these are the short fragments. These are the short fragments. These are the short templates. I told already. So it's about repetition. That's what I told already. The short templates are the major DNA. That means the DNA fragment with target sequence. Whereas the long templates contain extension. That is how the cycle repeats. This for up to cycle 3. Then you can repeat for 4 up to n times. I told you already. If we, at the end of the 32nd round, we can get as many as up to 1 million fold copies. This is about the schematic presentation. That means the first step, denaturation. That means we have to break down the DNA double strands. We will get single strands. Then at 95 degrees temperature. That is very important. Then the second step is called renaturation or annealing. Simply that means the primer will hybridize at the target at the appropriate region at 55 degrees centigrade. The third step is DNA synthesis. That means the DNA polymerase will extend the chain. That means from the 3' hydroxyl group of the primer. Then we'll get long template. This is cycle 1. Then the cycle does the same steps. The first denaturation, then the renaturation or annealing, then the third DNA synthesis. Then here we'll get short templates. This is very important. The short templates contain the original DNA fragment. Then the original strand, the long template, the short templates, all they participate together in third cycle. Likewise, it will continue up to n numbers as many as we can as per our desired requirement. Okay. This is about the procedure of DNA polymerase chain reaction. Okay. With respect to graph, for better understanding, we can draw a graph how it went well. For example, check graph for illustration. We'll take between, that means 3 phases versus temperature and time. This is temperature. This time in minutes. This is 50. This is 60. 70. This is 80. This is 90. This is 100. This is 1, this is 2, this is 3, this is 4, this is 5, this is 6, this is 7. Okay. Say the first step, what we call denaturation, takes place at around 95 degrees centigrade. Around 95 degrees centigrade. It will be around for 1 minute. Okay. After that, the second step, this is called denaturation. Denaturation. After that, the second step, what we call renaturation, somewhere around 55 degrees centigrade. Renaturation. It is also for 1 minute. Renaturation. And the third step is denaturation, so around 75 degrees centigrade. 75 degree. It is for 2 minutes. This is for 2 minutes. What we call DNA synthesis. And again it will continue. That means this is just a graphical representation. Graphical representation of PCR, between temperature and time. Say at around 95 degrees centigrade, the DNA strands will break up. What we call denaturation. Then temperature will come down around 55 degrees centigrade. What we call renaturation or annealing. Then again at around 75 degrees centigrade. That means the Taq polymerase. Here we have used the enzyme Taq polymerase. The Taq DNA polymerase. It will take for around 2 minutes. That means that around 3 to 5 minutes. One cycle runs for 3 to 5 minutes. So it's about the DNA, polymerase PCR reaction respect to graphical representation. Okay. Then what are the different variants of PCR? What are the different variants? This is very important. Being a versatile, I told you, it is indispensable molecular biology technique. Most of the experiments associated with PCR, it is like without PCR, like we want a photocopy, so we maintain a Xerox machine. In office, Xerox machine is essential because we want many photostat copies. Now, similar to that, we want many copies of respect to target DNA fragment. So PCR is like integral part of molecular biology technique. But there are different variants. For example, inverse variants of, it depends on the kind of demand. variants of PCR. Say inverse PCR, asymmetric PCR, RT-PCR, that means reverse transcriptase PCR, then real-time quantitative PCR, anchor PCR, then RAPD, random amplified polymorphic DNA, then AFLP, amplified fragment length polymorphism, then RACE, rapid amplification of cDNA ends. These are very important. You know, inverse PCR, asymmetric PCR, RT-PCR, real-time PCR. These are the abbreviations are very important examination point of view. So RAPD means simply the basic question: a random amplified polymorphic DNA. And AFLP, amplified fragment length polymorphism. And RACE, rapid amplification of cDNA ends. Okay, these are the both different variants. So depending on the kind of demand, we can use a PCR appropriately. Then the last topic of PCR technique is its applications. You know, the main objective of any technique is to generate as many applications of as it can. The most important application of PCR is clinical diagnosis. Clinical diagnosis. You know, it's like prenatal clinical diagnosis of inherited disorders. Then clinical diagnosis of retroviruses. Then clinical diagnosis of bacterial infections. Then the second important role is its role in forensic science. You know, there are too many applications are there, but I would like to emphasize a major important application in human health welfare point of view. Say, we would like to, as there is suspicion that if the womb, that means the growing womb, may have certain kind of genetic disorders. Maybe the growing kid may have some kind of genetic abnormalities. We would like to ascertain. Say there is a technique of extract the cells from amniocentesis, then subject the same to the analysis, but for that PCR is important. Say sickle cell anemia. This is for what we call prenatal clinical diagnosis of inherited disorders. Sickle cell anemia. Beta thalassemia. Phenylketonuria. This is one most dangerous point mutation. Sickle cell anemia we have discussed in the last class. At the beta gene, at the 6th position, the glutamic acid has been replaced by valine simply because of transversion mutation. A to AT, that's it. And we have a suspicion that we would like to ascertain the same. For that, PCR is essential. What we call prenatal clinical diagnosis of inherited genetic disorders. For example, sickle cell anemia, beta thalassemia, phenylketonuria. Then clinical diagnosis of retrovirus like HIV. For laboratory diagnosis of the outbreak of viral infections. not only for HIV, any other retroviruses require PCR. PCR amplified technique is most important. Then for my bacterial, for say mycobacterial tuberculosis, even for certain cancers also this is very important. Say virus induced cancer like human papilloma virus that cause cervical cancer, and even for that, but that means it is an indispensable tool for clinical diagnosis of virus causing cancers like HPV. And most important, forensic sciences, say what we call in criminal issues, say in criminal place where there is a minutest availability of sample which contain least fraction of DNA, even one single DNA molecule. And say maybe say one single DNA molecule from a semen or or from a hair, or any other body fluid which has been available in less adequate manner. That means if one DNA molecule we get from the crime spot, with the help of PCR we can as many as molecules as we can, DNA molecules, I mean the target DNA fragment. Say for example, I told you already, at the end of the 32nd round we can get 1 million fold of the original DNA fragment. That depicts the kind of the strength, that means quantitative and qualitative impact of polymerase chain reaction in the field of molecular biology. Okay, these are the good different applications. Apart from this, there are different another roles also there. Okay, these are the major roles. Okay, so this is for today, and in the next class we will discuss some other topic.